everyone. I would like to welcome everyone joining this evening from different locations of India to this unique program where we would like to understand and taste science with a different flavor. So to start this particular session, we would definitely like to have some panelists. The panelist for today is Dr. Dennis Messina, who is graduated with MSc in Immunology from Quebec, Canada, and he is Master in Public Health from John Hopkins Institute, Bangalore, Baltimore. With a 22 years of vaccine industry experience, he is currently leading the global medical team of Sanofi's pediatric segment of vaccines. So welcome, Dr. Dennis.
Thank you, and hello to everyone in India. It's a pleasure for me to be with you today.
Thank you, Dr. Dennis. On the other hand, we would like to welcome Dr. Suhas Prabhu, who is a consultant pediatrician at Hinduja Hospital, had been the faculty of various state, national, and international conferences, had been an author of many publications of international and national repute. Welcome, Dr. Prabhu.
Thank you. Thank you, Sumit.
So, in order to start this conversation, I think that would be a great idea if we would just have a look on a small documentary that we had prepared. So can we have a look on the documentary?
Pertussis or the whooping cough. A highly transmissible respiratory infection that continues to be a public health concern. Around the globe, up to 16 million people are affected by pertussis each year, with approximately 195,000 children dying from the disease. It wasn't until the 1930s that wholesale vaccines for pertussis became available in combination with diphtheria and tetanus toxoids. By the 1970s, massive vaccination coverage reduced the incidence rate from 157 cases to less than 1 per a lakh individuals. But in 1979, concerns with reactogenicity resulted in a decline in acceptance among public, leading to the ceasing of wholesale pertussis vaccination, and eventually the return of pertussis as an endemic disease. Administration of too many shots to children was another reason for reduced compliance among parents. However, with the introduction of combination vaccines, the burden of numerous injections got mitigated, and grouping of multiple antigens into a single injection helped increase vaccination coverage rates. The need for a safer vaccine led to development of a less reactogenic, acellular pertussis vaccine. Clinical trials demonstrated that the combined acellular vaccines were less reactogenic than the combined whole cell vaccines. By 1990, majority of the high-income countries had switched to acellular vaccines, leading to a sharp decline in pertussis incidence. Despite the evolution of vaccines, dosing and mishandling errors related to mixing antigen components before administration added complexity to the process. In 2013, Sanofi introduced a new fully liquid hexavalent vaccine, following the revolutionary concept of adding all the antigens in a single vaccine formulation, thus providing one-shot protection against diphtheria, tetanus, pertussis, poliomyelitis, Haemophilus influenza type B, and hepatitis B without reconstitution. The HCP and parental response have been more favorable towards this new way of vaccination, with higher satisfaction, increased efficiency, and less impact on daily life activities. A fully liquid vaccine saves about 5 minutes across 3 doses and has 5 times fewer mishandlings when compared with a hexavalent vaccine that needs reconstitution. These benefits make the Sanofi vaccine the most preferred vaccine among HCPs and the #1 acellular hexavalent vaccine worldwide, with global market share of 67%.
So, Dr. Prabhu, we have seen the development of combination vaccines. I think from the video, one thing is very clear that everywhere, adherence towards immunization and compliance remains a concern. And when it is complemented by complaints from the parent, it's definitely a tougher time. So, may I ask you, like, have you experienced and differences in complaints and compliance as we evolved from WP to AP vaccine?
Yeah, I think it's very important, Dr. Sumit. And what you showed in the video is what I've experienced.
Okay.
I've been practicing for 40 years now. So I've seen the time when the reactogenic whole cell pertussis vaccine was the only one that was available.
Right.
And I've experienced administering that to children. And I can tell you there has been a sea change over the last 40 years. And I used to literally dread giving the whole cell pertussis vaccine. If on a particular day I had to give it to maybe 3 or 4 children, in spite of counseling, I would be bound to be woken up later at night. The child doctor has got high fever, the child is crying a lot. And in spite of telling the parents all this, there would always be a concern in the parents.
I understand.
No parent wants to see their child cry.
Very true.
And I've seen that big difference now that has happened when we started using the acellular pertussis vaccine. So definitely the number of complaints have been very less. And if you ask about the question of compliance, yes, sometimes it would happen. I won't say it happened very often, but I remember at least a couple of patients who, after a severe reaction after the first dose, didn't come back for the second and third vaccine. And that used to happen, of course, very, very rarely. But yes, there was an issue about compliance because parents were very hesitant to give the second dose after having experienced the child's trouble and pain during the first. But you talk about compliance by parents. I'll tell you another story about compliance by doctors.
Okay.
I know at least a couple of doctors, one of them including one of my own relatives, who happened to give the whole cell pertussis vaccine. It was not a combination vaccine in those days. This is, I'm talking about 40 years ago, to his own nephew. And the child unfortunately had a seizure and had several complications, and he remained retarded for the remaining portion of his life. And that person stopped giving the whole cell vaccine to anybody. He said, if this can happen to my own family member, I would hesitate to give it to the next. So you're talking about compliance by parents. I'm telling you, even doctors were reluctant to comply with giving the vaccine, the whole cell vaccine.
It's so sad to hear. I think that is increasing the vaccine hesitancies. So
yes, I mean, that's a rare thing, but yes, it did happen. And unfortunate, but yes, it did happen. In my case also, I had a couple of patients who had seizures after following the vaccine, but fortunately, nobody ended up retarded or with any brain damage. But we know that that's could happen with the whole cell vaccine. I have never seen that with the acellular pertussis vaccine. Definitely a big change.
So thank you so much, sir. I think let's understand from Dr. Denny, what could be the probable reasons for such complaints which we had seen, and we had seen the sharp decline in acceptance of pertussis, whole cell pertussis vaccine from the Swedish study. What could be the reasons of the probable complaints from WP vaccine?
Well, as was just exposed very clearly, the difference in safety profile of the whole cell vaccine and the strong reactogenicity it provokes is a problem. Of course, febrile seizures, even probably leading to brain damage, is extremely rare, and that is a major concern. But yeah, those prolonged fevers, prolonged crying, do give hesitancy to the parents, and although maybe some experiences that it's rather rare, what data has shown globally is that it's actually something that truly affects the parents' willingness to bring the child back. As far back as the clinical trials that were conducted with the cellular vaccines, in the Cochrane collaboration meta-analysis, they clearly found that children who received the whole cell vaccines were 4 times more likely to interrupt the vaccination schedule because of their adverse events than those who had received the acellular vaccines. And now that's a few years back, but very recently in Brazil, they found that about a fifth fewer children who had experienced adverse events after the first dose of vaccine were up to date and complete on their schedule of vaccination because compared to those who had not experienced adverse events. So clearly, these adverse events bring hesitancy in the parents. And it's a very good point that was raised also earlier that HCPs played a role in that too. We have historical data from Italy, from Germany, where the low coverage rates that we're seeing, I mean, we were talking about 30%, 40% observed in the 80s in Germany and in Italy with DTWP. And the reason that physicians were citing for not promoting and pushing parents to vaccinate their children was the safety profile, the fear of generating adverse events that would require, more visits, etc., but also can cause concern, because you have to remember that, most of the adverse events are self-limiting, but they can, the prolonged fever can turn into febrile seizure, and it's hard to predict when. So, and in which child. So really, the safety profile is something that's important. But more generally, I think the combination vaccines, the hexavalent combination vaccines with a cellular component, do bring a breakthrough, technological breakthrough for this, because not only do you benefit from the acellular vaccine lower reactogenicity, but you also have a single injection against the 6 diseases. It's a win-win solution also for both the healthcare professionals and the infants and the parents. From the healthcare professional, it's more peace of mind that you're covered. You don't have as many injections. You have less psychological stress, fewer visits, less concern that the parents are going to come back and because of the complaints from the vaccination. And from the parents, it's also more peace of mind, because you know that your child is protected against all these 6 diseases without the risk of having adverse events from a whole cell vaccine, but also having the risk of adverse events with multiple injections. Really, as I said, in some historical examples that we have from, say, from Germany, showed that when they switched from the whole cell vaccine to the acellular vaccines, the VCR increased significantly. But also, once the acellular vaccine was established as a standard of care, they saw that with the implementation of combination vaccines with more antigens in them, the proportion of children who were completely vaccinated and vaccinated on time for all of these antigens increased. As an example, the polio and hepatitis B timely and complete vaccination almost doubled between the pentavalent period and the hexavalent period, clearly showing that there as well, the multiple injections, the reduction of these multiple injections also brings confidence to the parent, because obviously, this is something, regardless of the safety profile in the end, having multiple shots in a child is something that can be concerning, especially for new parents.
Well, thank you so much. I think, sir, that's very well said that, development of fully liquid hexavalent vaccine in AP formulation is not only helping in increasing VCR, but also assuring compliance of immunization schedule, because
Right.
one shot 6 injection is definitely a boon to the immunization schedule. So since we have understood the differences of adverse event, I would say, the complaints and improved compliance. Now, I would like to ask second question, which is specifically that we see 2 different set of vaccines. Have you seen any significant difference in the clinical profile, when you have vaccinated the data from the WP vaccine or AP vaccines in your practice?
If you're asking Sumit about the efficacy of the vaccine or the performance of that vaccine, then I mean, it's difficult for me to be certain. I don't keep longitudinal data on my own cohort of patients. Earlier, whom I immunized with the whole cell vaccine, and now in the last more than 2 decades, the acellular vaccine. But in my experience, I have never really seen a patient who has received the full course of either of the vaccines come back and say, "Doctor, my child got whooping cough. Why did he get whooping cough?" So I would like to believe, and I'm not going into the data there, probably Dr. Denny can give you the data. In my personal experience, if there is a difference in the seroprotection or the efficacy of the whole cell versus the acellular pertussis vaccine, it probably is very marginal and it's not really of great significance to an individual patient or to a doctor who wants to immunize in his own practice. Now, I would also think that for pertussis, for example, we talk about herd immunity. So when you say herd immunity, the coverage of the population with the vaccine is equally important or more important than the fact that whether this vaccine will give you a 2% better seroprotection or a 1% better protection and so on. And you saw the data, we all saw the video, you saw what happened to Sweden.
Right.
Because there was so much backlash against the wholesale pertussis vaccine causing adverse effect that the vaccine was stopped and people were not taking it. And that caused a greater problem than possibly a little bit of difference in protection with the acellular vaccine compared to the wholesale vaccine. So I think those are my concerns, and I am perfectly happy to use the acellular pertussis vaccine. And I think we also need to talk about the fact that, and maybe this can come up later in the discussion, that we also need to ensure that our children are protected from pertussis even at a later age.
Absolutely.
And we all know that the wholesale vaccine we simply cannot give after the age of 5 or 6 because of its reactogenicity. There's a lot of data now, even the Indian Academy of Pediatrics recommend that we give a booster at 10.
Absolutely.
So that has to be the acellular pertussis vaccine. So we don't really have an option. So this would be my take on the so-called difference between the protection rates of the acellular pertussis vaccine versus the wholesale pertussis vaccine.
And I think it's better to give positive experience from the start itself.
Yes.
That's great. That's great. So Dr. Denny, I would like to ask the subsequent question to you. With the advancement of these technological advancement with respect to AP vaccines, what makes a difference in the effectiveness of the AP vaccine as such? Do we have any data?
Well, for sure, the efficacy first of the acellular vaccine that are available today has been demonstrated since early on in the clinical trials. And as was pointed out today, WHO, for example, recognizes that adequate protection is achieved for complete infant vaccination with either acellular or wholesale. However, in terms of available data, it's important to point out that whereas the current acellular vaccines that you can find and that can be given to children today have been tested in clinical trials, but also in real world, and I'll come back to that maybe later. But I want to first point out that for wholesale vaccines, one of the problems is today's vaccines don't have any data on efficacy and effectiveness. For sure, and I agree with what I've said earlier, there's no evidence that today's the wholesale vaccines that are used today don't work, that they don't protect. You can maybe question a bit the quality of surveillance, etc. But still, if they weren't effective against pertussis, we would know it. We would see it with large numbers of pertussis cases, especially in young children who among whom you usually don't miss pertussis. So there's no strong evidence that they don't work. But what I want to point out, though, is really that there's a technological difference between acellular and wholesale that is very important. Wholesale vaccines are both technologically crude, but also biologically complex, right? It's the entire bacterium with minimal purification or control, essentially, on its composition. We don't know how many antigens and how much of each antigen is present in them. And what that translates into is and I'll give you examples of this, is that it's difficult to control and be sure of a consistent potency for these vaccines. Canada, Netherlands made painful experience with that. And outside just the Netherlands example, they had, they were manufacturing their own wholesale vaccine at RIVM, and up to the early 1990s, this vaccine was 90% effective. They did, but through the process, they changed minute changes, change of filters, change of component supplier for the culture media, things like this. And so minute changes in the manufacturing process. By the late 1990s, early 2000, the effectiveness of the vaccine had dropped to 30%. But all the while, that vaccine had passed all quality controls. what this illustrates is that it's difficult with whole cell vaccines to be sure that your vaccine will retain its performance over time, because it's hard to control it. On the contrary, when you take the acellular vaccines, you have a very precise and reproducible manufacturing process that ensures that you know precisely every time how much antigen is in there and how potent it is. And how this is illustrated is that you have the historical clinical trials that showed 85%, 90% efficacy originally in the 1990s. But as recently as last year or just before COVID, the data that was published on their effectiveness, real world effectiveness, was still above 95. Sweden published a study covering 2007 to 2018, and it was above 97% for the infant vaccination schedule. And I had the honor of presenting a study that we conducted in Mexico. Mexico's been using solely our Sanofi Pentaxim and then Hexaxim vaccine since 2008, and we measured the effectiveness of that vaccine. And over this 2008 to 2019 period, a 3-dose of the vaccine was 97% effective, and a 4-dose was 95% effective all the way to 4 to 6 years of age, when the child is supposed to receive a booster, bringing me to that point. You know, the data that's available today has been controversial. You know, if you followed a bit pertussis, there's been lots of controversies on the quality of protection, and I think some misconceptions have been put out there. Pertussis immunity after vaccination or even after infection is never lifelong. And the data that has accumulated, whether it is from Russia, from Iran, from Poland, Laos, and various countries using whole cell vaccine, has clearly showed that even after the whole cell vaccine, the protection wanes, and that after an infant series, a child becomes susceptible again by school entry. They need boosters, and they need boosters to. The importance of boosters is that, especially in school children, adolescents, these are populations that are very efficient transmitters and amplifiers of circulation, and will generate outbreak just by themselves. And so it's important to perpetuate protection in these populations, give boosters, even if the primary series is a whole cell series. So I hope this provides you some of the more global perspective on this, that clearly, you know, whole cell or acellular are protective, it seems, but there's more confidence with acellular vaccines. And I want to say, I'm pretty proud of the study we did in Mexico. It clearly shows that as recently as the last 5 years, very high effectiveness for Sanofi's acellular vaccines, contrary to what some people have said, as effective as any of the acellular vaccines that are out there, and clear need for boosters beyond infancy to perpetuate protection and ensure a community that the community is protected from broad circulation of the bacteria.
Perfect. I think Dr. Denny very well summarized, and I think Dr. Prabhu, you just said that, you know, for pertussis it is important that we need to ensure coverage.
That's right.
Irrespective of the vaccine. At the same time, the AP vaccines coming, as Dr. Dennis mentioned, that, you know, is giving more confidence to the clinician with the consistent vaccine effectiveness,
Right.
and that is a great boon. Now, since I think, sir, we have seen the development of AP vaccine, which is a complex process, but actually giving a consistent response. As a clinician, you always look for clinical and scientific data.
Right.
You know, I know, sir, because having an interaction with you all the time, so I know this. So, sir, as for you, like, you know, what are the scientific parameter, if I would like to just quote, whether it is seroprotection rate or geometric mean titers, or, you know, the safety of a co-administration? What are those parameters, like, you know, you usually see in a complex schedule of 1614, sir? And how would you like
it's
to
very important. You need to have scientific backing for saying that this vaccine works. And you mentioned at least two of the main parameters that we look at: the seroprotection rates and the geometric mean concentrations, or the geometric mean titers. Some people refer to it as GMT, following the vaccination. Now, I think the audience is pretty aware when we say seroprotection rate, it means the number of children who develop an antibody level above the minimum
Right.
required, usually 14 or 28 days after the vaccine is given. And it may be a single dose, it may be a series of doses or whatever. And then if it's, of course, 100%, it means every single child that you give that vaccine is now getting adequate protection from that particular disease. So this figure is important. The SPR is important because it tells me how effective the vaccine is in protecting the number of children that I am vaccinating or trying to protect. But this is only half the story.
Okay.
Because we speak about the GMC or the GMT. Now that tells us not just how many are above the required level, but how many times above the minimum level they are. So
Fantastic,
that's
sir.
the titer, that's the number or quantitative estimation of the antibodies that are being produced by this particular child after 2 or 4 weeks after the vaccine course is over. Now, why is this important? When we say that the lowest level is all that you really need for protection, why do I need a much higher titer? How does it matter whether it's 4 times the minimum or whether it's 10 times the minimum? Because this speaks about not only the robustness of the response, but also possibly the longevity or the duration of the response. We all know that titers keep waning over a period of time unless there is, of course, subclinical reinfection and re-stimulation of the immune system. And as Dr. Dennis just said, we know that there is not going to be lifelong immunity. And there is now some data that even after getting actual pertussis, you don't get lifelong immunity. So why do you expect it from the vaccine? So we would like to minimize the duration on the number of boosters. So when I have a vaccine that gives me very high GMTs, I am confident that my child is or my patient is protected for at least a period till his booster is due. So these two factors are very important, the SPR as well as the GMT. Now, also, I would also like to point out, I would like to have this data in my population. That means the Indian population. So you really cannot extrapolate data from other countries and say that it's likely to work here. We all know our experience of using the oral polio vaccine to try to eradicate polio.
Right.
It worked pretty well in so many countries, and we all know that story. Let's not get into that. That's not what we're discussing today. But we had to give 10 doses, 12 doses, pulse polios every year before it really worked. So we know that every population, every environment is going to react differently. So I would like to have data on Indian populations. That's the next thing, not just the SPR and GMT data, but SPR and GMT data on an Indian cohort. So that's the other thing that I would like to see. And you also mentioned about co-administration.
Yes.
Now, we know that in our country, we are now giving a lot of vaccines all at the same time. So this is what is sometimes referred to as density of vaccination. We give them all together, and we all know at 6, 10, and 14 weeks, we're going to immunize against not only these 6 diseases that you mentioned, which comes as this fully liquid acellular vaccine, but we also vaccinate for rotavirus, we vaccinate for pneumonia.
Absolutely.
And therefore, I would also like to know whether there is some kind of an interference or, when these vaccines are given together. So when I have that kind of data, it is reassuring to me that the vaccine that I'm giving is going to do its job, and it's not going to interfere with the other vaccines that are being given concomitantly. So that's the kind of data that I would be looking for. And I'm sure Dr. Dennis is going to give you that data to tell me that this vaccine is the one to use.
Yes. So question to you, Dr. Denny, could you please help us with the immunogenicity and safety data of fully liquid 6-in-1 vaccine that from the different schedule as well as schedule of 6 and 14 that we follow in India?
Yes, for sure. I won't get specifically to the data. There's one point I'd like to bring up again on these measures, because for sure both SPR and GMCs or GMTs are important. But the one thing that's important about them is that we need to make sure that these are robust measures, right? We need to understand how they're measured to understand the robustness of the evidence they provide. And one thing that where the technology of using acellular vaccine is also advantageous in that area is that we have precise antigens against which we can measure the immune response. Whole cell, it's a little bit more complex. Now, using, for example, antibodies against pertussis toxin is a good start. or against the same antigens as the ACLA vaccine more broadly is a good starting point. But they have to be measured in a precise and accurate way. And too often, this is really too bad, because too often what we see when I read articles about clinical trials or studies on whole cell vaccines, what I see is that they've used commercial ELISA kits. These kits are developed for the diagnosis of pertussis. They're not designed, validated to measure and compare immune responses to the vaccine. There's semi-quantitative assays, and so what we, it's hard then to be able to trust and be confident that whatever GMCs or seroprotection rate they provide is reliable. Contrary to that, typically ACLA vaccines, we've licensed it in Europe or in other countries. We had to essentially show that we had a validated assay that was reproducible and accurate in terms of its measure. So that is a question also on the data that you see, say, in the product label, for example. But to come back to your point, for sure, there's a wide array of different infant schedules around the world. There's 3 doses with no toddler booster, 3 doses for the booster, 2 doses for the booster, and even among the 3 doses plus booster, you have 2, 3, 4, 2, 4, 6, 6, 10, 14 weeks. It varies extremely. And so you want to make sure that a vaccine is tested across these different schedules to make sure it works as well in one and in another. And especially those schedules like 6, 10, 14, where there's only 1 month between the doses, can be challenging. It's long been demonstrated that for most of the infant vaccines, it's better to increase a bit the interval to 2 months, say, to ensure that the subsequent doses actually stimulate more strongly the memory and build up a strong and lasting immunity in the children. Now, when it comes to the 6, 10, and 14-week schedule, I want to say, as far as Exacxim is concerned, we're pretty covered around the world. We had a clinical development plan that has more than 25 trials conducted in Latin America, Africa, Asia, Europe, and in all the diversity of schedules, including the 6, 10, 14 weeks. That, I'm sorry to say, it wasn't in India, in the Indian population. Maybe one day we'll have a trial there, but for the moment, the study we have was in South Africa, where the schedule is 6, 10, 14 weeks plus a booster at 18 months. And there, the seroprotection or seroconversion rates for pertussis and for all the other antigens were from 93% to 100% after the primary series of 3 doses and 94 to 100% after the booster dose. And that was consistent with what we've seen in other studies where a 3-dose, 1-month interval schedule, such as 2, 3, 4 months in Turkey, for example, or even with the 2-month interval schedules in 2+1 Nordic countries or 3+1 in Latin America. So clearly, consistently across the schedules, we've seen a very strong performance. So there's no doubt in my mind that having seen that across continents, population schedules, this consistent performance of the immunogenicity of the vaccine, I'm confident that if today we were to conduct a clinical trial in India, we'd find as high seroprotection or seroconversion rates and GMCs with our Exacxim vaccine. Now, to your point on co-administrations, for sure, it's important to show that, as you were saying, the schedule is getting complex with more and more vaccines, some of which can absolutely not be combined. So we already have a hexavalent there with Exacxim, and it's important to test that immunologically you don't have an interference, but also that it's safe. And from that point, I can say that Exacxim has been tested with co-administration with pneumococcal conjugate vaccine, with rotavirus oral vaccine, with measles, mumps, rubella, with varicella, and with meningococcal vaccines, at least with the conjugate A, C, Y, and W. And we're actually in the process now of finalizing a clinical trial to measure and test the co-administration with MenB, which is becoming or being rolled out in some countries as yet another additional infant vaccine. And in all of these trials that we've conducted, both immunologically and from a safety point of view, no signal of interference or higher reactogenicity was observed. So we're confident that the vaccine can be administered with all of these.
Well, thank you. I think, Dr. Prabhu, just wanted to give you the perspective that registration studies has been done with the fully liquid hexavalent vaccine in India, and where we had seen the seroprotection rate is ranging from 94 to 100% with the complement of high number of geometric mean titers and concentration. So I think that is on one side. Since and one important aspect which is actually giving the birth to the next question. In India, we are following 6, 10, 14 weeks, which is anyways a challenging schedule, at the same time complex schedule, and with assurance of birth dose of hepatitis B. So probably that is the schedule that we are having. So I would like to ask you that what is the importance of birth dose of hepatitis B in India, and if missed, how we can ensure that it is there with the infant which has been missed? How to ensure that it is
okay? I mean, let's get to the important point. We know that the purpose of giving hepatitis B vaccine is to prevent a carrier status of hepatitis B virus, which we know later on can cause, cirrhosis and liver cancer, of course, at a much later age in life. And we know that the incidence of becoming a carrier is higher the younger you are when you acquire the infection. Therefore, early protection becomes very important, because we know that, for example, in the first few weeks or month of life, if the child acquires hepatitis B, there's almost a 90% chance that he's going to become a carrier. And that's the importance of giving a zero dose. We also know that if the mother is hepatitis B carrier, then of course the zero dose is very important. And of course, additionally, we also want to give the immune globulin at birth, but that may not be freely available in the periphery. But even then, the birth dose becomes very important. Like you rightly mentioned, there are instances when the birth dose is missed. It's part of the national immunization program. And till today, I think that a large majority of births in our country do happen in either private maternity home or in a public hospital, where they would ensure that the hepatitis B dose is given at birth. But Dr. Denny also mentioned something very important. For hepatitis B, when we first started giving the vaccine, if you remember, Dr. Sumit, the recommendation was 0, 1, and 6 months. So the increased interval is very important to ensure full seroconversion.
Absolutely.
So the big question comes now, and we know that in the US, for example, it's 2, 4, and 6 months, and we want to give it at 6, 10, and 14 weeks. Of course, for programmatic reasons, it becomes much easier to give it at And also in our country, we know that we have these diseases at a younger age. We can't afford to wait till 2, 4, and 6 months to immunize against pertussis, for example. If we follow that schedule, we're going to have much more pertussis in young infants. So we have to give 6, 10, and 14. So now the question comes whether when you give 6, 10, and 14 weeks, hepatitis B, now I'm talking about the hepatitis B component of the 6-valent vaccine, are we sure that our children are getting fully protected? And as I mentioned to you earlier, it's the most important thing to see that they're protected early. We can't wait for that booster dose at 1.5 years. So we need data on whether this fully liquid hexavalent vaccine given at 6, 10, and 14 weeks, which is a compromise. The recommendation was actually earlier 0, 1, and 6 months. How effective it is in protecting against hepatitis B, and especially if the zero dose has been missed. Now, if the zero dose is given, then we are giving 4 doses. So hopefully the seroprotection will be better. But as you said, and I also mentioned, there will be instances when that is being missed for whatever reason.
Very true.
And even today, I see patients come to my clinic and I find out they were delivered in a maternity home in Mumbai and still didn't receive the hepatitis B for whatever. So this question becomes very crucial. When I give 6, 10, and 14 weeks hepatitis B, how well is the protection? How good is the protection for hepatitis B? And I would be very happy with this fully liquid hexavalent vaccine, which contains hepatitis B. That's got good data, which says that over 95% or 96% protection is there, which I don't think is there for some of the other vaccines that is available. The figures are not as good as this. So that's also very reassuring to me that in case the child has even missed hepatitis B dose at birth, my 6, 10, and 14 weeks is also going to protect him for hepatitis B.
Very true, sir. I think let's hear from Dr. Denny. may we
Yes.
have, like, what is the immunogenicity data of, fully liquid hexavalent acellular pertussis vaccine with respect to hepatitis B?
Mhm.
What is that data? Like, could you please help us?
Sure. I mean, of course, the hepatitis B immunogenicity has been tested across all the clinical development of Exacxim in the various studies. I want to focus maybe on the data from one study, the study I mentioned actually before in South Africa. Why? Because it is that schedule that is the same as in India. And um, what in that study, what we opted to actually test is exactly the situation that you're referring to, where you standard maybe to give the hepatitis B vaccine at birth, but it may not occur because of a reason or another. So that study was actually designed with two groups. One group that received the hepatitis B at birth, a group that didn't receive hepatitis B at birth, and then both groups received Exacxim at 6, 10, and 14 weeks of age. Now, of course, the group that received the hepatitis B at birth, after 3 doses of Exacxim, had received 4 doses of hepatitis B vaccine and had higher anti-hepatitis B GMCs, around 1900 milli-IU per mil, than the group who didn't receive the hepatitis B at birth, and therefore had only received 3 doses, and the GMC was about 330. So lower, but what's important is that in both groups, the seroprotection rate was 99%, 95, 96% respectively, which means that almost all of the children were protected. And that's what's important in that interval, because we're talking about short-term protection there. We want them all to be protected for that interval. Then they all received the booster dose at 1.5 years. So that constituted a 5th dose in one group, a 4th dose in the other group, and the same difference in GMCs was seen. And but still, seroprotection rates were extremely high: 100% for those who had received hepatitis B at birth, 98.5% in those who didn't receive hepatitis B at birth. So it's still almost all of them protected after the receipt, 1 month after the receipt of that toddler booster. But of course, that brings the question: is that what does that difference in GMCs mean in terms of the durability of the protection? And we did actually 2 studies, one in Thailand and in one so one was in Thailand, one was in Europe, where we actually tested the durability of that protection by giving, at 6 years in one study, 9 years in the other, a hepatitis B vaccine challenge to measure how strong the anamnestic response was, as a proxy measure for the memory that the infant vaccination had elicited. And while in the 2 studies we had 54% at 6 years, 49% at 9 years of seroprotection, just before the challenge. 1 month after the challenge, seroprotection rates in both cases had risen to above 90%, 97%, and 93% in the other at the 9-year interval, meaning that essentially almost all of the children had restored protection as soon as they were challenged, meaning that if they had to encounter hepatitis B, they were all protected 6 to 9 years even, and maybe beyond, for that, which means that even that horizontal transmission of hepatitis B that may occur in the first few years of life, which is minimal but still part of the risk, is there. And I want to point out also a study that is not yet published, but will be presented at ESPID this year in May, which is an analysis that was done in South Korea, where hepatitis B at birth is a standard, especially for women who are seropositive for hepatitis B, or their infants rather. And we worked with some researchers there to analyze, is there a difference in risk if instead of the 016 standard schedule, you integrate hepatitis B in the hexavalent vaccine, so therefore into that infant vaccine schedule, and therefore there's a slight delay in the second dose. And actually, we found that the study did not give any rationale for an increased risk of delaying from 1 month to actually beyond 8 weeks, so beyond 2 months even, the protection. What is essential, the vaccine. So what is essential, what that means is that the essential piece in protecting young children from that vertical transmission in the first few weeks and a couple months of life is that birth dose and potentially the immunoglobulin. But delaying to 6 weeks or 8 weeks, the second dose of hepatitis B did not have any impact on the risk of acquiring hepatitis B infection.
Well, thanks. I think, Dr. Prabhu, I was just referring to the data from, from WHO that, the official estimate of birth dose in India is less than 75%.
Yes.
And I think over here, as Dennis has also mentioned that, although with the hexavalent, fully liquid hexavalent that was given in South Africa, we had seen that although the birth dose is given or not, seroprotection rate is achieved, but the geometric mean titers were double than those which are
Right.
actually the otherwise standalone vaccine. Not only this, I think I would also like to quote that in India we have seen the seroprotection response with the hepatitis B birth dose along with hexavalent vaccine. We have seen zero response of 96%. I think that is what I would like to
Right.
give. And as he mentioned, anamnestic response has also found to be very robust when the children were challenged.
Right.
So I think with that, it's very important that we need to stick the vaccine which actually gives a good seroprotection rate, which is complemented by good geometric mean titers as well. Well, I think thank you so much, Danny, on this. But let me just move ahead with another question. Now I'm going to ask you a very practical question, sir. And the question is that we have moved from in the previous monovalent combination vaccine to fully liquid vaccination.
Right.
So, sir, have you seen any reconstitutional error during your practice, and how impactful is a fully liquid vaccine in your point of view as a clinician?
Even doctors are humans. We can make errors. And sometimes it's the nurse who prepares the vaccine. They can make errors too. And unless they are supervised, you won't even know that an error has been made in reconstitution, in inadequately drawing it from the vial, or sometimes the reconstitution put in the wrong vial, or simply you forget to put something. It could happen.
It could
probably happen. So you're very right in saying that once you have a fully liquid reconstituted vaccine in your hand, the safety becomes much higher, that nothing is missed out. The full dose is given to the patient, and the other error that you can happen, that might happen, don't happen at all. So I would be very happy. And I think you have data. I've seen studies from Korea and other countries. We said you showed that in your video too, and over 96 or 97% of healthcare personnel would want to use a fully liquid reconstituted vaccine so that it's for safety. But besides safety, I would like to mention two other things. I think one of the points was also mentioned in the video, is that the time that you save.
Correct.
So I think we are consultants. Sometimes a patient who comes, and it doesn't happen now. In the old days, they would say, "Doctor, where is the medicine?" I say, "I've written on the medicine." And sometimes the parent feels, "What am I paying you for?" He's paying me for my time. So the old saying is that the time is money. So if I save time in doing something, then yes, that's money for me. But it's not just the fact that speed and saving of time. There's also the issue of the ease of doing it. When you actually have to reconstitute, you have to pay attention to what you're doing. Otherwise, you're likely to make an error. And I had the experience that when I was doing that, you would, for example, need to concentrate. So you'd sometimes have to turn your back to the patient.
Right.
You lose eye contact. You lose the eye contact with the parent. And I think all that is very important when you actually practice to build up your practice. So ease, safety, and speed of use from all 3 points of view, getting a fully liquid reconstituted, and that too ready-made, prepared in a syringe. There's nothing like it. And as you showed in the video, all healthcare practitioners would definitely would prefer that.
Thank you so much. So quickly, Danny, any global perspective on fully liquid acceptance from yours, and quickly, we are anything which you would like to add?
Sure. I'll be very brief because the subject was already well covered by Dr. Peru, and with the introductory video that you showed. I mean, we have conducted studies on this. Obviously, technological advance of formulating 6 valencies into fully liquid is not just intended to put all the vaccines in one as such. The purpose is really ensuring that we increase the combination, and the increasing combination of antigens translates into a quicker, safer, and more precise act of vaccination, really. The study in Belgium, which you showed some results of, clearly demonstrated, you save half of the time, or your preparation time is cut in half, but you also reduce the risk of errors by 5 times. Errors such as missing reconstitution, but also sometimes non-aseptic handling of the vaccine in the syringes. It's sometimes hard for healthcare professionals to admit this. And it was interesting, the study we conducted in South Korea that was just referred to, because there, anonymously, the healthcare professionals, nurses, and physicians were asked to answer this. And the candid answer we obtained was quite interesting. It showed up to 52% of HCPs report having missed reconstitutions or made other preparation errors with vaccines that require reconstitution. And whether it's the data from South Korea, but also we had surveys conducted in France, in Germany, in Italy, there's a clear preference for physicians and nurses for a fully liquid formulation. And the reasons are exactly those: the quicker administration, which allows more time for care and counseling too. We were talking earlier about making sure the parents come back for subsequent doses, having a full vaccination. Well, that's part of it, counseling and making sure that parents are aware. The quicker the vaccination, the more time on this. Safer administration with less risk of needle stick injuries or non-aseptic handling, and the more precise active vaccination, reducing the risk that you miss reconstitution, you miss some antigens, and in the end, the infant is not fully vaccinated against all 6 diseases.
Well, thank you so much. I think it's now all these features are making that the vaccine has been accepted in various countries with 67% of market share. I think with that, we are at the end of our session, sir. We are running out of time as well. So I think with that, I would like to conclude. And thank you so much for coming over here and sharing your clinical experience with us. Thank you, Danny, for sharing your viewpoint as well as the data which are coming for a fully liquid hexavalent AP-based vaccine across the globe, and giving us the confidence that we should be carrying in order to take probably a step towards increase VCR and increase public acceptance with an effort to enhance SCP's efficiency. So I think with that, I would like to conclude this fireside chat. Thank you so much for joining in, and see you.
Thank you.
Thank you, sir.
Thank you, Dr. Denny.
Thank you very much. It was a pleasure. Have a nice evening.
Thank you.
Thank you.